Layo yung mic. Sandali lang. Okay, there it is. Hello everyone. We are back with another another science episode. And today I'm going to talk about oncogenes and tumor suppressor genes. So hello George and Joyce. Welcome back to the stream. Magandang gabi po sa inyong lahat. Ako po si Bini Millennium. Ang ika-nines? What's that? Sa lo at imaning pitolo. Ika-siyam na member ng Bini. Okay, so, yes, right now, let's see. So, if I do the jumping jacks now, it's gonna be the ninth set. So, as a reminder for if there are newcomers here, it would be, so we recently hit 12,000 subscribers. Do not mind the fact that it is now 13. It was at the 12,000 mark that I did, like, that I made the post. So it's 12,000 subscribers, and I was like, why not, to celebrate, do 1,200 jumping jacks? Okay? And I was like, I don't think I'm built to do all of those in one go. So I was like, divide it into around 10 sets, so it's still 1,200. And so far, we've finished the first, second, third, fourth, fifth, sixth, seventh, and eighth set. Right? Wait, let me, one moment. Where are my notes? I think, yeah, we finished the eighth set. Yep, it was yesterday at the 1 hour mark. So, that means I've done 8 sets of 120, 16, and that's 960 jumping jacks. That's a lot. But why not do the 9th set today? Because, why not? And the biggest proof of that is the heart rate monitor. Please don't mind the fact that it's already at 115 with just me talking. That's normal. That's the normal resting heart rate of an Isekai'd hero who got put on Earth and has a lot of things going on in their head. Okay? Now let's do the jump jacks. The 9th set. Alright, let's go. Let me just move this really quickly. I think that this one works. Yeah, it does work. Okay, let's go. Road to 3K. Miles or kilometers? Doesn't matter. Just the goal is just not to die at the end of a marathon or a triathlon. One of those. Okay, let's go. And 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30. Wait lang. Ano muna? Breathe muna. Okay. And 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46, 47, 48, 49, 50. Okay. So that's 50. Among asthmatic ginaasma na. But we are strong soldiers. That was 50? Yeah, it was 50. Wow, look at the heart rate. It's like going. 50. And one more time. 51, 52, 53, 54, 55, 56, 57, 58, 59, 60, 61, 62, 63, 64, 65, 66, 67, 68, 69, 70. Wait lang. Malayo pa lang water. 70, 70. But let me just type it somewhere here. 70. So I don't forget. And 70, 70, 70, 70. And go. 71, 72, 73, 74, 75, 76, 77, 78, 79, 80, 81, 82, 83, 84, 85, 86, 87, 88, 89, 90. 90. Wow. 90. Just a little bit more. Oh, why are the bouncies not working? The bouncy thingies, where are they? It's gonna be the stream buddies. Well, stream elements. It depends if it works there or not. 90. Wait a second. Wow, it's 158. There, found it. Okay, 30, 20, 30 more. Okay, go. 91, 92, 93, 94, 95, 96, 97, 98, 99, 100. Okay, so for the last 20, I always do this. I count in Chinese for the last 20. Because why not? That's how life works. Let's go. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Okay. The world is spinning, but we are here on Earth. So that's 120. Oh, will it hit 160 heart rate? Hopefully not. Well, okay. So, we officially finished. No, 120. The 9th set of 120 jumping jacks. Whoop whoop, and that makes it to 160, 120. Oops. 080, 1080. Oh my gosh. Jumps. So, yes. Let me just use this one. One moment. Whoop whoop. Right now, we are going to talk about ONCOGENES and tumor suppressor genes. So, this one is a very interesting topic because it's about cancer. And we know it's the big C word, big cancer word. So what is cancer? I will draw a mini map of it, okay? Oh, wait, this one works. So for cancer, let's start here. Cancer is a lot. One more set to go. Yes. And then if I'm regarding the set, I'm actually debating if I can add an 11th set because just the other day we reached 13,000 subscribers. So if it's 11, look at this, times 11, 0, 2, 1, 0, 2, 1. It's gonna be 1320. And just a mini add-on. Highs there. So the things I say on stream, it's funny. Yeah, but we are finished 9th set. Whoop whoop. And about cancer, we have 1, 2, 3, 4, 5 mini definitions for it. So cancer is a collection of diseases. It is uncontrolled cell growth. That's the very common term for it. It is also involved with inhibited cell death. Cell death, unlimited replication, and genetic mutations. So, if someone asks you, what is cancer? It's a collection of diseases, inhibited cell death, uncontrolled cell growth, unlimited, oh, this is a bit similar, the unlimited replication and uncontrolled cell growth, and genetic mutation. Essentially, imagine if Naruto can just times Kagebunshin Jutsu forever and no one would stop him, he will just occupy the whole earth with the Taju Kagebunshin Jutsu. Alright? So, the bigger the amount of time, the more mutations. So, in a drawing form, it would be like this. Let's see. Like, wow, where is it? Okay. So, if you have normal cells, these are just your normal cells, innocent cells, and then you get a precancerous lesion. Just more cells like that. And then it becomes stage 1, which is still localized tumor. Stage 1 is localized. Stage 2, what is it? It's early, localized, but advanced. Wherein you kind of have your tumor here, and then some blood vessels start beside it. And then you reach stage 3. Well, hopefully not you, but this thing over here is reaching stage 3. You have your tumor here. This is the late stage, starting of the late stage. And then you got stage 4. The metastasized, it will really spread already, and it's really exposed to the other stuff, and it will just go and attack the other organs or spread essentially. Is there anything wrong with these cells in particular, or is it just uncontrolled growth? Half, because some of these cells are like the useless. Well, let's say a cell that can't function, right? Normally, it would be, I'll draw it in different colors. Normally, these cells, let's say, I'll make it red, the bad ones. The normal working ones are green, okay? Let's say the red one is the first cell that we should get rid of. It should be degraded. The reason why it should be degraded is because, let's say it has a mutation, it's not functioning well, or let's say it's doing something else that you want. But let's say it spreads more and it's not destroyed, then the green ones will not be able to function properly because the red is just overcrowding it. So that's why there could be something wrong with them. Most of the time there's something wrong with them, or they're just not functioning properly, and they're also in a state where they just overpopulate the whole thing, and your normal functioning cells cannot function anymore because it's just too crowded. Just take note, all of these, the reds and the green, will want the same food, quote unquote. So if they're fighting for the resources that your body has, then let's say this is the burger, this food, then there won't be enough food for everybody, and your green stuff will not be able to function well if they're also competing for the same resources as those cancer cells. So that's hopefully that answers your question. But yeah, as for the risk factors for cancer, I have about 4 here. So let me just draw it out here. So regarding the risk factors, okay? Risk factors for cancer. You have the predisposition. where disposition. This is genetic factors and mutations. So sometimes I'll talk more about genes, but sometimes it might just be is a sleeper agent the right word? Where it's like hidden, and then at some point it'll just happen. Or let's say it wasn't supposed. Let's say that sleeper agent was supposed to never be like woken up, but then with certain environmental factors, for example, environmental factors, you smoke, or you don't wear sunscreen and are just super exposed to sunlight, or you eat a lot of sadly you eat a lot of spam, chorizo, tocino, hotdog, and the fat part of the lechon kawali. If you have the mutation for it and you combine it with the environmental factor, then you're a big winner. Okay, it's like a 1 + 1 = boom. And there's also the behavioral factors. Factor. I agree. It is indeed delicious. So that's why I don't eat it every day at least. Maybe every other day. Yeah, but for the behavioral factors, it's smoking and obesity. So try to maintain a healthy weight in a healthy way. Do not go on crash diets, because you might become thin suddenly, but then you'll just regain it super quickly with a little bit more stuff. So just normal exercise and normal food. Number 4, health conditions. That would be viruses and inflammatory disorders like diabetes. So try to not hit all of these. It's not a good bingo to be bingo. Because let's say a lot of your family members have, like, let's say a lot of them have, like, or they're related family members. Well, you are related to your family. A lot of them like had specific type of cancer and stuff like that, and you're related to them. And then you smoke, and then you don't wear sunscreen and just go out and do sunbathing, and you also eat a combination of chorizo, tocino, and then you are also not controlling your food, and thus you're obese, and then you unfortunately are also diabetes. Bro, you don't want to be a big winner and have all of these. Okay, so try to spot that something that you could fix. The one that could be really fixed would be where is it? Red marker. Don't smoke. Wear sunscreen. Do not do spam every day. And try to lose weight. And then if you are pre-diabetic, try to not reach the number for diabetes. Boom! Hopefully you'll live a little bit longer. Yeah. Okay. So for the modular approaches of how to not fix it, I don't know, but like the steps of it. Oh wait, let's do it one more time. Yeah, if you don't go outside, take note. There are the light bulbs are also a concern. Well, so that's why. But because they are still UV, they still have UV. So that's why I watched some documentaries of like K-pop idols and K-drama actresses and actors who also wear sunscreen even if they stay inside. Just to make sure. Skincare. #skincare. So yeah, risk factors again, predisposition, behavioral factors, environmental factors, and health conditions. Yes, indeed, skincare. So for the modular approaches of it, it is modular approaches. You have risk factor assessment. You have diagnosis. It just like early detection. If you think or feel that something is wrong, go to the doctor. Do not say, "Oh, I'm just okay." It's like no, go get checked, because it's gonna be harder if it's in the late stages and you're like, "Oh, maybe I should visit the doctor." The doctor's like, "Why'd you come just now, bro? We could have fixed it in a cheaper and faster way." But now that it's in super late stage, it's a bit harder. So just get checked. And then for prognosis, this is checking the like tumor sequence, like at what part are you and how you can like treat it. That's the fourth one. This is the targeted theory. I know that's true. So just try to not get sick. But that would be also hard. So try to not smoke or drink or wear sunscreen. Summary, wear sunscreen. Yeah. Treatment would be targeted therapy. There. So risk factor, diagnosis, prognosis, treatment. Okay. Oh, that one. We could use this emote. That one works. So next, where is my page? I had a page for it. So again, what is cancer? Yeah, that's funny. But yeah, again, what is cancer? It's a collection of diseases, uncontrolled cell growth, unlimited replication. What's the fourth one? Cell death. You cannot inhibited cell death and genetic mutations. Now, there is a certain experiment called the RSV experiment here. RSV. RSV. So this is the what happened to the letters? RSV. This is the rose sarcoma virus experiment. This is the experiment that showed that cancer can be caused by a virus. Like this. Cancer can be caused by a virus. A virus. It just means it's a specific type of virus. It's the I don't know how to say this. Sarcoma virus. RSV. So now you have, this will be a beautiful drawing. You have a chicken. Like this. Like this. And the tail. And the legs. Beautiful chicken. So this chicken, the manok, has a tumor somewhere here. Let's draw it. Let's say its tumor is here. Chicken. Let's say that's the tumor, right? Now let's say you remove the sarcoma. Remove the tumor. You just chop the chicken, get it. And then you slice it into small pieces. Now, don't slice the chicken. Just get the thing that you need. You ground it up into smaller things, and then you pass that tumor into a very, very small filter that's designed to, let's say I'll put the red stuff here. A very, very small filter where you subtract or remove the cells and bacteria. So only the virus is here. And then you collect the filtrate and put it on a young chicken. An innocent young chicken. You inject it there using the injection. There. That's the injection. That's not a firecracker. And after a while, the injected chicken, actually, this is the chicken. The injected chicken actually develops that tumor in the same spot as the old chicken. So that work laid the foundation of like, there are oncoviruses aka cancer-causing viruses. Wait, there's paper that fell. Got it. And one more. I got it too. Okay, so this one laid the foundation that there are cancer-causing viruses. And also the foundation for a thing called whoa, letters. This one. Word. There. Laid the foundation for a thing called oncogenes. Yes. AKA genes that drive cancer when they are mutated or exposed. Alright? So if you zoom in on a very, what's this called? Molecular level, this is how it's gonna look like. It's just, so that was the chicken version, kind of in vivo. Now we're gonna do in vitro, like the tubes and all. So this is the transformation process. Okay, just as a reminder, what happened to the chicken? You have a chicken with a tumor. You get the tumor, you chop the tumor, you inject those, you put it through a filter so you remove the cells and other stuff. You only have the virus now. You inject that virus into your young chicken, and the young chicken develops the tumor at the same spot as the old chicken. Boom. Proves that okay, that this we discovered oncoviruses. Now this transformation process is now this one. Let's say you have a plate, a plato, and then you have the cell monolayer cells. You inject this with your RSV particle. So another injection like that. You inject it with the RSV particle. And then the virus introduces oncogenes into the cell, and then you see it grow like this, slowly but surely. Wait. You see your thing grow more like that. And some fibroblasts, or like cells, lose the contact inhibition, aka normally if a cell hits the end of your plate, the edges, I mean, they will start to die, and that's called contact inhibition. And also if they start to touch each other. But then some, in a lot of tumor cells, there is a loss of contact inhibition where they will just continue to times two, even if they're already in contact. So that's why you would see a dense cluster of cells growing on top of each other. All right. And then now we have, where is it? I have a page about oncogenes specifically and tumor suppressor genes. But let's just go through the what's this called? The properties of transformed cells first. Where is it? Found it on this page. Identifying this figure. 1 second. Just getting the paper. Okay. Now, for the properties of transformed cells, how would you distinguish like a cancer cell from normal cells? We need a table. Table. Nice. Okay, we have cancer cells and normal cells. I talked about it a little bit before, especially regarding its contact inhibition, but this is just these are just more details about it. Okay. So you have altered morphology, like this. Altered morphology. Cancer cells tend to be a bit round, background. Normal, flat. There's the loss of contact inhibition. What is it? Cancer cells will just continue to multiply even when they're touching, but normal stops dividing. Stops dividing. They are all, in terms of anchorage, independence, cancer cells can grows in suspension. Normal cells need to be attached. Need to be attached. Needs to be attached. Plural, attached. Singular. Need. One. And then immortalization. You can guess that cancer cells can activate the telomerase, and that would prevent the telomere shortening, aka allowing unlimited cell division. So telomerase is super active. Meanwhile, normal cells, they would undergo replicative senescence, a little bit of aging slowly after a certain division due to the telomere shortening. What is telomeres? Those are the ends of your DNA sequence. And remember how your cells normally multiply or divide? They divide to multiply. And then as they do that, your telomeres would just slowly shorten because it can't be completely perfectly copied. Alright? And now, where was I? There is also a reduced requirement for mitogenic growth factors. Mitogens? Question mark. We talked about mitogens earlier, where normally, let's say, a mitogen would start to go in. This is about the mitogen P53 and P51, P21, okay? Mitogen would attach to a thing that's very similar to your RTK. It would call in, your RAS-GEF, that one, it would be affected, and then it would be activated, and then it would, okay, I'm gonna write it, mitogen, and then your RAS-GEF, and then your RAS is activated and phosphorylated, which would then go into the RAF MEK ERK, aka your MAP kinase pathway. This is the MAP KKK, MAP KK, and MAP K. That kinase would then phosphorylate some factors such as the things that would then trigger your P53 to be active. P53 would then go into the nucleus and would trigger some transcription and translation of your P21 protein, which then helps the cell cycle to stop. Was it? P21 would help it stop. One moment, just verifying. Stops your cell cycle. Yeah, it's in apoptosis. It halts your cell cycle. It stops cell cycle progression. So if this is screwed up, then they can just multiply a lot. So that's why cancer cells are like, I don't need mitogens. I will just multiply because I want to. Now, there is high saturation density, very saturated because they can just be super dense. Meanwhile, the normal cells would want to be a little bit flat. Inability to halt proliferation in response to growth factor deprivation. Okay, so even if you lower the growth factors in cancer cells, they will just continue even when there's lower nutrients, while normal cells normally would want food. Glucose transport. Glucose and metabolism. So they exhibit the Warburg effect, where they prefer aerobic glycolysis even in O2 rich conditions. Means higher glucose uptake, which supports rapid growth because they just want to multiply. And there's also wow, this word, tumorigenicity. When injected into mice, immunocompromised mice, transformed cells form the tumors, aka mice that have a lower immune system will form the tumors. And now, those are just many qualities of cancer and normal cells. Now, about your RAS versus a MIC, okay? RAS versus MIC. RAS is a GTPase. And it is involved in pathways like the MAPK and PI3K, AKT. We talked about it a little bit, not a little bit. We had about 2 to... Hello, Mafuran. Yes, times 2, Kagebun Shinjutsu. Oh, I should introduce myself. Hi, I'm Millennium Isekai'd Mask Knight Empress Scientist. Sometimes I do my intro as magandang umaga, gabi, hapon, madlang people. Ako po si Bini, Aya, Mika, Stacy, and all of the other members. Essentially, Bini Millennium, the 9th member of Bini. Thank you very much. The MV is out. And we do Pokemon, Pal World, Science. I will be doing Bio Inc. just to review the human systems. Was it scheduled for later? I think, yeah, later. So I hope I can finally save a few patients with that one. And then I think in a few days I will just try to relax a little bit after a couple more science streams with the Cafe Nipponica, where I will try to run a Japanese restaurant. So feel free to stop by. If not, there are VODs and shorts, funny VODs and funny shorts, and funny mini clips here in the channel. So yeah, about the RAS versus MIC. Oh, I forgot to include the jumping jacks and the heart rate monitor and the shiny hunting in Pal World and Pokemon. Yes, I forgot to include that, but now they're included. Voila. So RAS and MIC, okay? Those are different things. RAS is a GTPase, and it's involved in pathways like the MAPK and AK3. We know that. RAS, oh, I have a mini flowchart. RAS, RAF, MEC, ERK could also be PI3K, AKT, and MTOC. I will just draw it again in a bit for the page. Meanwhile, MIC is a transcription factor. It is activated by ERK, transcription factor. And then if you activate it, it will then regulate more genes for the cell growth and all of that. So MIC, if you have MIC and RAS together, it can, well, if you screw up MIC and RAS at the same time, bro, that's bad. So look at this one. This age, and this is tumor-free mice, aka mice that don't have the tumor. So if you screw up MIC, it's just kind of slowly like that. This is MIC. Oh, wait, 1 second. Cheeseburger. Yes, hello, Sleepy. Oh, welcome to the stream. I think is this your first time gone by? Introductions again. Hi, I'm Millennium, Isekai'd Mask Knight, Empress Scientist. Sometimes the 9th member of Bini Millennium. Sometimes I do Pal World, Pokemon Science, and Jumping Jacks and Heart Rate Monitor and Shiny Hunting here. But right now we're talking about cancer, because why not? I need to get through this one. Yeah, and just remember, if you have RAS and MIC right now, RAS and MIC, RAS is a GTPase involved in certain pathways like the MAPK, and then RAS as well. Now, if you screw up MIC, okay, fine, Fred. If you screw up MIC, the age of those, you get the older, well, you have less tumor-free mice, aka they just got more tumors, okay? That's MIC. But here's the funny thing. It's not very funny, but here's an interesting thing. Oh, I'm gonna use a different color for the slightly colorblind people. If you screw up RAS, the tumor-free mice lessens more is your RAS. Why? because RAS is involved in more pathways compared to MIC. Because if you look at it here, the RAS is kind of more of the starting point. MIC is somewhere at the tail end a little bit later. So that's why if you screw up MIC, it's a little bit less of an effect compared to if you screw up RAS, it's gonna be here. And here's the ultimate plot twist. I'm gonna use purple because red plus blue is what? Purple. Violet. Violet is more bluish. Purple is more reddish. Yeah. So if you screw them both, if they are both not at a good condition, burn, your mice will just not exist anymore. Well, they will get tumors at a very higher rate if both your MIC and RAS are dysfunctional, dysregulated. If they are not working well, that's the term. Okay, so hopefully we just remember to avoid smoking, wear sunscreen, maintain your weight, do not eat spam, chorizo, and tocino every day. Split them up. Just MWF instead of Monday, Tuesday, Wednesday, Thursday, Friday. If you really want to. Just try those. And maybe do jumping jacks 120 every other day. So now about ONCOGENES and tumor suppressor genes. The title of this video. Because the page, nahulog yung page, but now I found it. So about ONCOGENES and tumor suppressor genes. You can have overactivity mutation. Never smoked, so good. Okay, continue that. Do not start or think about it. Just hunt shinies, because if you do shiny hunting, that will take about 6 to 24 hours of your day. And that means you won't even have time to think about those things. Just set alarms for food and rest. Very good, very good. You don't want to get cancer. Try to not drink as well, because that's gonna screw up a lot more stuff too. And if you are also pre-diabetic, or don't even think about crossing the diabetes number, because that's gonna be rough. It's harder to lower it down a little bit if it's already hitting the number, but it's still possible. Okay, so about the overactivity and underactivity mutation. Oh, very good. Don't think about the drinking. Drink water, I mean, but not the drinking. Right? Now, for overactivity mutation, that is your ONCOGENES. AKA, let's say you have a normal cell over here. Like that. Normal cell. And then you have a single mutation event. Okay, single mutation. It just takes a single mutation event, and like here, let's say here, and this will now be glowing. It will now be an active mutation, which enables the oncogene to promote cell transformation. And that will start to screw up your RAS, your MIC, and HER2. Take note, RAS causes the RAS pathway will just screw up a lot of things, like your MAP kinase and all of that. MIC is involved with something called lymphoma, and HER2, any guesses? It's involved in breast cancer. So yeah, it's involved in breast cancer. Now, you have underactivity mutations. Okay? That is a loss of function. So here, for the underactivity, you have a normal cell again. Normal cell. You have a mutation event which inactivates it. I'll just do ME. We don't have space. You have a mutation event which inactivates the tumor suppression, the TSG. But the second one will activate it. So now both of them are affected, and boom, Two inactivation events would eliminate your TSG. And it's in the name. A tumor, tumor, tumor suppression gene. If you eliminate it, then they cannot suppress that tumor. So that's where your tumor, the P53 is involved, because if it's affected, then they cannot stop your cell cycle from cycling, from continuing the cycle. Tumor. Tumor. There you go. Wow, that's a weird word. Tumor. Oh wait, tumor. T50, the P53, and then you also have the RB1, which controls the G1 to S, aka like greater cell cycle again. And then you have the BRCA1/2. Any guesses where this is involved? I think this one's a more commonly heard one. Tuesday. Tommy and a Tuesday. Bottle of water. Just say tubig, the water. BRCA1. That is involved in ovarian and like breast cancer as well. So it's a homologous recombination repair. Mutations in it increase your breast cancer, the chances of getting breast cancer. So there is a thing called, okay, just mini review, overactivity versus underactivity. Overactivity, just one mutation can activate the oncogenes, and some oncogenes would be, for example, your RAS, your MYC, your HER2. Screw up MYC, it's a little bit bad. Screw up your RAS, incredibly bad. HER2 also, breast cancer. Underactivity mutations, you need two events to knock it out. You need to punch it twice. In PAL world, that would be rocket launcher and Jet Dragon doing its missile on it. So that's P53 and the RB and BRCA. Now there is a term called, let me see, one moment, mutual exclusivity. Mutual exclusivity. Where? Okay. It means the tendency of certain oncogenic mutations to rarely co-occur in the same tumor. What does that mean? If you already have like a mutation in your KRAS, wait, the best example would be, I'll give more examples, but there is this, are these mutations just super rare, or do they regularly happen but your system fixes it? Good question. Very good question. Okay, I vaguely remember the answer, but I'll try. Every day, I think mutations are common. They just happen, but your body has the DNA repair mechanisms to repair them. But in the unfortunate event where the repair mechanisms screw up, which is normally a very rare occurrence, then that's when the slippery slope happens. And those situations where your DNA repair mechanisms screw up might increase in the event of, let's say you always just go, not just outside, let's say you always do like sunbathing without sunscreen, then your chances of getting skin cancer might be just a tad bit higher compared to if, let's say, you kept wearing sunscreen even if you kept doing the sunbathing or going outside. So that's why, like, before the sunscreen was like a famous fad thing, like, and then people would just go outside without it, and then there's like a period of years where skin cancer was a common one, and then that's when skincare slowly started becoming an important thing. And so that's why like the environmental factor plus the genetics plus your food and your obesity stuff would play a role in it. Because for like yeah, because let's say you have a sleeper agent gene, but then let's say you're eating healthy and all that, then maybe it won't wake up, and then your body will at some point fix it as well. So yeah, just try to not get the 2-hit combo, or 3-hit or 4-hit combo especially. So for mutual exclusivity, that again, that one is the oncogenic mutations rarely co-occur at the same time, and that's because of a thing called functional redundancy. Redundancy. And once a key pathway is activated, additional mutations provide no further advantage. So, let's say you have, like, there's 3 examples here: the KRAS and the BRAF mutations, which both activate your MAP kinase pathway, the MAP/ERK one. You don't normally see them together because, like, especially like in colorectal and lung cancers, because a single mutation is enough to drive the tumor progression. That sounds sad though. Now, you also have your RB1 loss and cyclin D1 amplification, right? So RB1 normally inhibits the E2F transcription factor, while cyclin D1 would normally, okay, because if you lower your RB1, you increase the E2F. So cyclin D1 overexpression is unnecessary because it's already happening. And the example that I like is the TP53 and MDM2. MDM2. P53, what is it? It would, if it's screwed up, it's a very direct mutation where affects your P53 and then P21, and then your cell cycle. If this is eliminated, then your cell cycle will just continue, right? MDM2 causes excessive degradation of P53. So one is like your P53 is mutated and can't function well, so you don't produce P21 and cell function just keeps on going. MDM2 causes the destruction of your P53, so they're leading to the same thing. Both lead to P53 and activation, so they're already happening. It's a functional redundancy. So that's why there's the thing called mutual exclusivity, where the 2 mutations that would lead to the same thing rarely co-occur in the same tumor. Alright, I think I mentioned how to identify the oncogenes and tumor suppressor genes earlier. Oh, there it is. Oh, there's a part here about the human and CRISPR and agar assays. So I'll talk about it. One moment. Alright, you have the human and mouse cancer cell lines. We usually use this to study oncogenes and tumor suppressor genes. And then you also have CRISPR. What is it? Clustered regularly interspaced short palindromic repeats. CRISPR. So they help identify oncogenes. How? By overexpressing their candidates in oncogenes. But for tumor suppressor genes, you knock them out. Okay? For soft agar assays, you test. Yes, CRISPR. Whoop whoop. One of our favorite words, aside from Wi-Fi Chan. For soft agar assays, those would test the anchorage if your cells would still grow on, like, while attached or not attached. Xenografts would be implanting the human cancer cells inside immunodeficient mice. And then you have genetically engineered mouse models. Okay, you have a mouse. Mouse. Mouse? Mouse? Mouse. 1, 2, 3, 4. Beautiful, that's a mouse. You have genetically engineered mouse models. So, you can knock in, you can put, let's say you have a gene, you can knock in the models and mimic the oncogenes. Or you can knock out a certain gene and mimic the the tumor suppressor gene effect. You also have the organoids, like 3D cultures derived from cell tumors, which promote a more physiologically relevant system. Because so you can have stem cells, right? But that's 2D, but you can have organoids 3D model 3D, right? And in a 3D in a 3D model you can see more. You can also use computational tools or datasets. RNA-seq would check which of the genes are upregulated, and also if it's TSG, if it's silenced. Single cell sequencing would help track the tumor heterogeneity, which guide your therapy development. So here's a question. How would I test if two oncogenic mutations are mutually exclusive? That looks like a French cat. It's a very, very specific description, but I think I agree. If it is a mime cat, then it's gonna be like that. No, it looks like a cat that belongs in a prison. Bonjour, mademoiselle. Un, deux, trois. Wait, counting in French. Un, deux, trois. Cats. I cannot say the number 4. Really, it's a bit hard to pronounce for me. 5, 6, 7, 8, 9, 10, I think. I think that's 1 to 10. Counter check. Thank you very much. The spellings are weird. You can also count to 10 in Japanese. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Thank you, Naruto, and the tales for what's that called, for being there in my childhood and teaching us how to count 1 to 10 because of the tales. Because of the QB. Now, for testing if you have 2 oncogenic mutations that are mutually exclusive. So oncogenes come from proto-oncogenes. Cat bone feeling, yes. Those proto-oncogenes normally regulate cell growth and survival. So, gain-of-function mutations turn them into oncogenes and drive the uncontrolled mutation. Tumor suppressor genes, if you suppress them, normally it prevents proliferation, and you need 2 mutations to activate it. What is mutual exclusivity? It is the concept where two of those things that cause the same thing would not normally exist. The key mutations rarely occur in the same tumor, and a single mutation is enough. And an example would be TP53 and MDM2. How would I test it? Check computers, computation analysis, and that would help test if the 2 mutations occur. And I can do cancer cell line study, aka use CRISPR, clustered regularly interspaced short palindromic repeats, and introduce the single and dual mutations, and such as like check the proliferation of apoptosis and pathway activation. Just as a review of how CRISPR works, because I kept saying it and I have to review it now. You have normally, okay, let's say you have a document. You have this document, right? No, that's a picture. That's a water picture. This is a notepad. Here you have correct, and then you have wrong thing here, wrong thing again here. Now, if you want to correct this and this Then in the human need CRISPR Cas9. That is, you have the Cas9 protein, which is doing the cutting, the scissors. And then you have the guide RNA, which will hunt down the guide RNA. Scientists make that. Let's say you have on your gene, this is the wrong thing, this is the squiggles. This one will be the partner sequence, AATCG, because it's opposite. As your protein, as your Cas9 protein, oh my gosh, here. As your Cas9 protein goes like that, it would see this and it would cut this part, and then you can replace it with something else. So that's, or just remove it to silence the thing, and you would check this. That's how you introduce a single and dual mutations, and then check your proliferation. You can also check soft agar assays for the anchorage dependent growth, and you can do genetically engineered mouse, the beautiful Von feline, where you could knock in, but the oncogenes knock out, test the TSG function. And if you can also treat the models with targeted inhibitors and compare the single versus dual conditions. So if the cells with both don't have increased resistance, then ME holds true. Again, if the cell which has both your oncogene and your TSG happening, if they're the same level and not increasing, then your mutual exclusivity theory holds true. So that is what's up with your oncogenes and tumor suppressor genes, specifically in cancer. Yes! Whoop whoop, we finished! So just as a mini summary, we talked about what cancer is, the oncogenes, like the one hit versus two hit. Oncogenes only need one hit. Tumor suppressor genes you need two hits for it, and then it would be quote unquote inactivated. And then what else? We talked about the rule of the wall something, sarcoma something. The Rouse sarcoma virus, where you would have you would have a tumor chicken, get tumor from it, chop the tumor, put it through a filter, and then inject it on a healthy chicken. And then you would get a healthy chicken, not a healthy chicken, a new chicken with the tumor. Transformation process is similar to that. And then the differences between a cancer cell and a normal cell, where altered morphology, one also has loss of contact inhibition, where your cancer cell will just keep dividing even though it's touching the other cells already or the edge of your plate. And that cancer cells can grow in a suspension compared to your normal cells. Alright? And I think, and we did 120 jumping jacks again. We are now 1000 something. Whoop whoop. Thank you so much for watching, and see you next time. Bye.